Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, we are going to start today the immune response and its types in different types of immune response, right? So immune response, it basically is of two types. First is the primary immune response, and the second is the secondary immune response. Okay. Primary immune response, the moment the antigen or a pathogen comes in contact with the body. After an initial contact with pathogen or antigen, there is no formation of antibodies. The formation of antibodies starts after a lag. After a period of several days, the antibodies they start to appear. Okay. So at the initial or the first contact of pathogen or antigen, there are no antibodies which are present. Okay. Slowly the antibody titer is basically mean a unit. Okay, when it is a amount or a concentration, so there is a gradual increase in the antibody titer, okay, concentration, and among this gradual increase, the first antibody which appears is IgM and then IgG. Okay, so the first antibody which appears is immunoglobulin M and then immunoglobulin G, which is followed by a gradual decline in antibody concentrations or the titer. So it is like this. The primary immune response says what? The antigen enters the body. The body is not knowing that antigen, so it is not capable enough to produce antibodies. So there is a lag of many days in which the body try to adapt to observe to isolate the antigen, and after that the antibodies they start to appear, in which IgM is followed by IgG, and then the gradual decrement is seen in case of antibody concentration or titer. Then secondary immune response, the memory cells which are produced after the encounter of antigen in the host immune system, they can be lively for decades. Okay. Every new encounter with the same antigen results in rapid proliferation of memory cells. Okay, so every time the memory cells they are produced, they are proliferated. Okay, this is known as booster response. This is known as what? Booster response. The antibody concentration titer is far greater than the primary immune response. In secondary immune response, the amount of antibodies which are formed, okay, their concentration and their specificity to fight against a particular antigen is much more in comparison to the primary immune response. And the secondary immune response is basically aided or mainly aided by IgG antibodies. So primary immune response, it was first started by IgM, then followed by IgG, and secondary immune response, it is mainly of IgG. Okay. The above response is known as what? Secondary immune response or the animalistic response. Understood? Antibodies produced during secondary immune response have higher affinity towards antigen. They are more specific and they have higher affinity towards antigen. They are more specific. More specific. Understood? Secondary immune response includes mainly of spleen, lymph nodes, tonsils, Peyer's patches of small intestine, and appendix. So secondary immune response includes organs like spleen, lymph nodes, tonsils, Peyer's patches of small intestine, and appendix. It basically includes the secondary lymphoid organs. The secondary lymphoid organs. The secondary lymphoid organs, they are mainly associated with the maturation of lymphocytes. But they helps in maturation of lymphocytes. Maturation of lymphocytes. Along with, they help the lymphocytes to make a complement. Okay, so The acquired immunity basically is induced in secondary lymphoid organs. Okay, so this is the immune response, the primary immune response, the secondary immune response. Primary immune response is slow, IgM followed by IgG. The titer increases and then decreases. Okay, the lag period where no antibodies are formed. Okay, it depends upon the pathogenicity, mean the effect of the antigen. Okay, the secondary immune response, it comes up with the development of memory cells. Every time memory cells they develop when the same antigen attacks. Okay, so it is not like that memory cells, once they are formed after encountering the antigen once. No, the antigen enters so many times that so many times the memory cells are formed. Understood? So the amount of antibody in case of secondary immune response is much greater than the primary immune response, and that is why secondary immune response is very helpful to fight against the infections. Understood? It basically involves the organs like spleen, Peyer's patches, tonsils, means these are all secondary lymphoid organs. Understood? So this power of secondary immune response, okay, and to produce the immunogenic cells very fast, that becomes the basis of vaccination or the acquired immunity, the process of immunization. Understood? Now, this is the primary immune response and secondary immune response. You can see the primary immune response. This is the first antigen exposure. Okay, when the antibodies formation is not there, no antibody formation can be seen. Okay, so this is the second antigen exposure or the start of second or secondary immune response, you can see. So during the primary immune response, there is a lag in which the antibodies are formed. Okay, it first increases, okay, and then decreases. Then there is a lag, okay, then increases, then decreases. So mainly the IgM, okay, it initiates in primary immune response and then which is followed by IgG. So this part of immunogenic memory, okay, this is known as what the primary immune response. So during primary immune response, the IgM, it comes first and then IgG. Okay, then it decreases. Then the IgG levels increases to peak, increases rapidly in comparison to IgM. So this is the primary response and the secondary response. Understood? Now coming to next, line of defense. There are different line of defense, especially three line of defense in the body. The first line of defense is, it includes the physical barriers and the physiological barriers. All these barriers, they are already being discussed with you. Okay, I am just revising this topic because the question comes again and again. The line of defense, the first line of defense includes the physical barriers and the psychological barriers. Okay, physiological barriers, sorry, physical barriers and the physiological barriers. Okay, secondary immune response, it includes cellular barriers and cytokine barriers. Okay, it includes cellular barriers and cytokine barriers. And the third immune or the third line of defense, it includes the acquired immunity or the adaptive immunity. So first line of defense is what? The physical barriers like skin, mucous membrane. Okay, the physiological barriers. The second line of defense includes cellular barriers. Okay, and cytokine barriers. And the third line of defense, it includes the acquired or adaptive immunity. Understood? Now coming to next, we will talk about the antigens. The antigens and their types. What are antigens? Antigens, they are the substances which when introduced into the body, they stimulate or they induce the immune response. They stimulate the production of antibodies, right? Chemically, the antigens, they can be proteins or they can be polysaccharides or both. Okay, or they can be nucleic acids. So chemically, antigens, they can be proteinaceous in nature, they can be polysaccharide in nature, they can be glycoproteinaceous in nature, or they can be of nucleic acids. Okay, mean RNA or DNA. Okay, so nucleic acid includes RNA or DNA, RNA or DNA, or it can be glycoprotein. Structure. Antigenic determinants every antigen is having many antigenic determinants. These antigenic determinants are known as what? Epitopes. They are known as what? Epitopes. They are the components of antigen. Each antigen carries many epitopes. Each antigen carries many epitopes. The antibody, this part has already been discussed before. The antibody, which is Y-shaped, okay, is having at least two antigen binding sites, okay. These antigen binding sites, they attach to a specific epitope in the antigenic determinant or an antigen, okay. Sometimes antibody can bind to the identical epitopes of two different cells, okay. So it can bind the identical epitope of two different cells at the same time, which can cause the neighboring cells to form a aggregate, okay, which forms the neighboring cells to form a aggregate, okay. Antigens combine with the antibodies. Aggregate means the precipitation, okay. Antigens, they combine with antibodies. So antigenic determinants is the part of the antigen which will attach on the antibody. The part of the antibody which will receive the antigen is known as what? Paratope. The epitope meets paratope. The epitope, paratope, they just fit like a lock and key mechanism. Okay. So each Y-shaped antibody molecule is having the two binding sites that can attach to a specific epitope or an antigen. An antibody can also bind to identical epitopes of two different cells at the same time to just neutralize the neighboring cells, to aggregate the neighboring cells. Okay. Antigens, they combine with antibodies. Understood? Antigenic determinants. So this is epitope, which is the part of the antigen which is responsible for its antigenicity, and this is the part of the antibody which will receive the epitope. So this is known as what? Paratope. Understood? Rest antibody, heavy chains, light chains, constant regions, variable regions, Fab, FC, when the antibody binding site, crystallizable site, everything is discussed with you. Okay. Types of antigens, complete antigens. There are two types of antigens, complete antigens and incomplete antigens. So complete antigens, they basically produce the antibody formation. They induce the antibody formation by themselves. Okay. When they don't need help of any other molecule or a protein to start the formation of antibody. Haptens, they are the partial antigens. They are incapable, they are not capable enough to produce the antibody formation by themselves, and that is why they need some other proteinaceous structure, other helper molecules to induce the antibody response. Okay, so haptens, they are the partial antigens. They cannot form antibodies by their own. That's why they need a protein molecule to help them, to assist them in the formation or induction of antibodies. Understood? These antibodies, or these are the protein carriers which help to form the haptens, or which help to form the antibodies by haptens. Antigens present on the body's own cells, they are known as what? Self antigens. And antigens which are not present on the host, they are known as, or they are regarded as non-self antigens. Okay. So self antigens and non-self antigens. Self antigens, they are autoantigens, and other are non-self antigens. H antigen is a precursor of A and B antigen, which is responsible for determining blood group A and B. Blood group A, B, and O. Okay. So H antigen is universal antigen. It is present in all human beings having the ABO blood group system. Okay, H antigen is universally present, and that is why H antigen, it doesn't create or doesn't remark itself, especially in the antigenic test. Okay, Bende et al. Bende Bende 1952, Bende and his company in 1952 from Mumbai reported a very special blood group in which all the antigens were absent. H antigen, A antigen, B antigen, all were absent. And he counted that blood group as Bombay blood group or OH blood group. Okay. So Bende in 1952, he basically really explored a blood group in which A antigen, B antigen, and H antigens were absent from the RBCs, and they were they or he called that antigen or that blood group as Bombay blood group or OH blood group. Understood? A very special properties of the individuals having the Bombay blood group or OH blood group, that they can only receive blood or they can only accept blood from their own group. Okay, because they have the antibodies against A, B, and H, and that is why they cannot accept the blood from the other groups. Okay, now coming to next, the complete antigens, incomplete antigens. Okay, monoclonal antibodies, that's a very important topic, okay, which was left actually to discuss previously. Monoclonal antibodies. Monoclonal antibodies, there are two types of antibodies production. One is the polyclonal and the other one is the monoclonal antibodies. Monoclonal antibodies, they are produced in vitro using tissue culture techniques. Okay, so with the help of tissue culture techniques in vitro, in lab, okay, these monoclonal antibodies are produced in vitro using tissue culture techniques. Okay, so MAB, monoclonal antibodies, they are produced by immunizing an animal, often a mouse, multiple times with the specific antigen. Okay, the same antigen is introduced again and again in a mouse, and the mouse will produce the antibodies. Okay, so it is a process of immunizing an animal again and again. Now B cells from spleen, the spleen cells, they are now they are taken out, and the B cells from the spleen, they are taken out. Okay, they are removed. Okay, now since normal B cells, they don't have proliferative capacity, they cannot proliferate. Okay, they cannot convert into any other type of cell like the prime cells and the effector B cells. So these B cells, they are provided a proliferative capacity by fusing them with immortal cancerous cells. See, there are two properties, that the cancerous cells, they cannot make antibodies. B cells, they can make antibodies, but they cannot proliferate. So we have just put both the characteristics in one category, that we have fused the cancerous cells which are able to produce the cells proliferation. They can do proliferation, but they cannot produce antibodies. And B cells, they cannot proliferate, but they can produce antibodies. So this fusion of cancerous cells with the, these cancerous cells are the myeloma cells. Okay, with the B cells is known as what? Hybridoma cells. They are known as what? B cells called myeloma cells to yield hybridoma cells. It is a mixture of B cells plus cancerous cells. Cancerous cells are also known as myeloma. So myeloma plus B cell becomes hybridoma cells. All of these cells are then placed in a selected medium, then a medium is taken for their growth, a cultured medium is taken that allows only the growth of hybridoma. Okay, when that allow only growth of the hybridoma cells, mean the cells which are till unfused myeloma cells and unfused B cells, they cannot proliferate, they cannot grow. Okay, so unfused myeloma cells cannot grow, unfused B cells, they cannot grow. Only hybridoma cells can grow. The hybridomas which are capable of growing continuously in culture while producing antibodies are then screened for desired monoclonal antibody. Okay. Now hybridomas, they have the characteristics of both. They can produce antibodies as well as they can proliferate. Okay. So now they are selected or they are screened to make the commercialized monoclonal antibodies. Those producing the desired monoclonal antibodies are grown in tissue culture. Now they are taken into tissue culture. The culture medium is harvested. Then the culture medium is harvested periodically. Okay, when the cells which cannot produce MAB further, they are screened out, and the cells which are continuously producing the antibodies, they are taken, and then they are purified. Okay, they are purified. They are extracted, purified. Okay, this is a very expensive and time-consuming process. So, in comparison to polyclonal antibodies is totally a procedure which is done in a living organism. Okay, and that is not expensive at all. Okay, so the monoclonal antibodies, they are it is very expensive and time consuming, but it is very specific. Okay, it is very specific. So it may take weeks of culturing and many liters of media to provide enough monoclonal antibodies for experiment or to treat a single patient. Monoclonal antibodies are very expensive, and that is why it's we are culturing both polyclonal and monoclonal antibodies wherever is required. Understood? So monoclonal antibodies, they are produced in this way. The procedure is simple. I will again tell you with the help of this diagram. The antigen is injected in the mice. This mice will produce antibodies. The spleen cells, these spleen cells are taken out. In these spleen cells, the B cells are there. Now they are fused with the myeloma cells. Now these are the cells. They are moved to a medium. Okay, some of the cells which are fused, they will only grow. The cells which are not fused, they will not grow. Only the hybroma cells, they will grow. Okay, now they after the growing of these hybroma cells, they are further screened to produce antibodies. If they are producing and desired antibodies, then they are screened in different steps, and they are used for commercialization. Understood? Now coming to next, applications of monoclonal antibodies. First is the diagnostic application. It helps in the detection of proteins of interest either by blotting or immunofluorescence. Immunofluorescence is a very important technique. Okay, like fluoroimmunescent hybridization technique is used in case of biotechnology. Okay, so monoclonal antibodies, they are used in various diagnostic procedures to detect different types of pathogens, bacteria, to diagnose pregnancy, okay, allergies and diseases such as hepatitis, rabies, okay, cardiovascular diseases, deep vein thrombosis, okay, location of primary and secondary metastatic tumors. When for the early detection of tumors, they can be used. And to trace the metastasis means the distant spread of the tumor cells. And now these monoclonal antibodies, these MABs, okay, they are fused with T cells to you know uh, to be used in immunosuppressive therapies, drugs. Okay, especially the kidney transplant. The T cells, they produce cell-mediated immunity to provide rejection against the kidney transplants, and these transplants can often become very, easy to go because of these MAPs, monoclonal antibodies. These monoclonal antibodies, they help to, decrease the T cell mediated immunity. Okay, they can control the T cell mediated immunity so that the person can easily have. Okay, so pregnancy testing kits, they are also produced by monoclonal antibodies. Okay, so therapeutic applications are to produce or to have radioisotope immunoconjugates. It is not of your use. Okay, uh, just you remember the diagnostic applications, rather the other applications are not of use in the detection of the toxin and drug. Immunoliposome based kits. Okay, and in cancer, I mean higher advanced diagnostic kits, they are used. Okay. Now, the next is antigen antibody complex. I mean, how antibody came into an action. So, epitope and paratope. So, there are some common terms which you have to remember. Agglutination. Agglutination is what? It is the clumping of microorganism, okay, or blood cell. Whenever a microorganism, means the epitope of the antigen comes in contact with the paratope of the antibody. Okay, compatible paratope of the antibody. Okay, the antigen antibody reaction takes place, and this is known as what? Agglutination. Okay, it is the clumping of microorganism in the microorganism goes non-functional. Okay, then opsonization. Opsonization is the process of making the phagocytosis of antigen antibody complex or the complement system easy. Okay, so the antigens, they are coated by antibodies, and these antibodies are known as what? Opsonins. These opsonins, they coat the antigen, and now this antigen is presented to different types of cells like macrophages and other phagocytic cells. Okay, precipitation. The next is precipitation. The phagocytic cells, they ingest the agglutinated microbe. Agglutinated microbe is in the epitope paratope combination of microbe and the antibody. It is the agglutinated microbe. Okay, it is being basically decorated by opsonins. Now the phagocytic cells like neutrophils, like macrophages, like mast cells, they are coming to engulf them. Okay. Soluble antigens come out of the solution. Soluble antigens, they come out of the solution and form a more easily phagocytized precipitate, which cross-linked by antibodies. Okay. So soluble antigens, they come out of the solution and form a more easily phagocytized precipitate when cross-linked with antibodies. Neutralization, the reaction of antibody with antigen blocks. When antigen comes in contact with the antibody, it blocks and neutralizes some of the bacterial toxins and prevents attachment of the some viruses to the body cells. So these are four terms: agglutination, opsonization, precipitation, and neutralization. Agglutination means epitope, paratope. Opsonization is decorating by the opponents so that they can be easily identified and phagocytosed. Precipitation is these basically phagocytic cells. They precipitate or they ingest the agglutinated microbes. Okay. When these agglutinated microbes they are ingested, the soluble antigens they come out very easily. Okay, and they are then cross-linked by antibodies. Neutralization is inactivation of any toxin of bacteria or a virus. Okay, when antigen comes in contact with the antibody. Okay. So, my dear students, these are some common terms which is to be addressed very importantly. Now, coming to primary and secondary lymphoid organs. Okay, the lymphatic system. Lymphoid organs are the places where actually the maturation proliferation of lymphocytes takes place. So there are two lymphoid organs, two types. The primary lymphoid organs. The primary lymphoid organs like bone marrow and thymus. Okay, so the primary lymphoid organs are the organs where T lymphocytes and B lymphocytes mature and they acquire their antigenic specific receptors. So I just write here the primary lymphoid organs, the primary lymphoid organs. I will not write organ, it's understood. The primary lymphoid organs, okay, so they come up with the maturation of T lymphocytes and B lymphocytes, maturation of T and B lymphocytes, along with antigen specific receptors. So in primary lymphoid organs, the T lymphocytes and B lymphocytes they matured and they are represented or they come up with antigen specific receptors. Understood? After the maturation, the lymphocytes they go to secondary lymphoid organs. Okay, and after reaching to the secondary lymphoid organs, they come up with further acquired immune response. Okay, they undergo proliferation and differentiation. So in the primary lymphoid organs, they are of two, bone marrow, thymus, and fetal liver. Fetal liver is also a primary lymphoid organ. Okay. So bone marrow, thymus, bone marrow, it helps in the maturation of B lymphocytes and T lymphocytes. Like in final maturation of T lymphocytes, it happens in the thymus. Okay. So bone marrow and thymus, they both helps in the maturation of T lymphocytes and B lymphocytes. Or we can say the primary lymphoid organs, they actually help to produce the immature lymphocytes, and these immature lymphocytes they are further matured. They are further matured. Okay, in these primary lymphoid organs, but they become the part of the acquired immune system when they reach to the secondary lymphoid organs. Okay, the secondary lymphoid organs it includes spleen, lymphatic vessels, lymph nodes, MALT, which is mucosal associated lymphoid tissue, gut associated lymphoid tissue, bronchus associated lymphoid tissue, skin associated lymphoid tissue, and nasal associated lymphoid tissue. So almost all types of mucus associated lymphoid tissue are mentioned, like gut associated, nasal associated, skin associated, and bronchus associated. Along with mucosal associated lymphoid tissue, it includes the adenoids or the tonsils, lymph nodes, Peyer's patches, okay, Peyer's patches, lymphatic vessels, and spleen. Okay. The lymphoid organs, two types. Okay, primary lymphoid organs and secondary lymphoid organs. I have already explained the primary lymphoid organs. Then the secondary lymphoid organs. The secondary lymphoid organs they come up with the maturation of B and T lymphocytes. Okay, they undergo proliferation, further proliferation, and differentiation. And differentiation. When we can say the primary lymphoid organs, it is concerned with the maturation of B and T lymphocytes, and secondary lymphoid organs, it is concerned with the proliferation and differentiation of B and T lymphocytes. The acquired immune response to antigens, okay, is usually developed, evolved in these organs. So, acquired immune response develops in these organs. The acquired immune response develops in these organs. Okay, so now these B and T lymphocytes, they become the effector cells. They become effector cells. Okay, in primary lymphoid organs, the lymphocytes, they get matured, they come up with the antigenic receptors, and the secondary lymphoid organs, they get proliferated and differentiated, and now they become the effector cells. In secondary lymphoid tissue, the lymphocytes, they actually move from one lymphoid organ to the other lymphoid organ through blood and lymph. So the process of migration, it is provided in secondary lymphoid organs. They become migrated, or they become migrants. Okay. So now the types of secondary lymphoid organs, what are the different examples of secondary lymphoid organs? Lymph nodes, lymph nodes. Lymph nodes are small solid structures found at small solid structure which are present in along with the lymphatic system. I mean, they are the part of lymphatic system. Part of lymphatic system. They are the part of the lymphatic system. They are basically composed of lymphoid tissue. They are composed of lymphoid tissue. Composed of they are composed of lymphoid tissue. Okay, and they act as filters for the lymph, preventing. They act as filter for the lymph, which is the tissue fluid. Okay. So lymph nodes, they are small solid structures which are the part of the lymphatic system. They are mainly made up of or composed of lymphoid tissues, and they help in the filtration of the lymph. Okay. The filtration of the lymph prevents, different foreign particles to enter the bloodstream. So lymph nodes, they also produce lymphocytes. They also produce lymphocytes. Right? And plasma cells. They also produce lymphocytes and plasma cells. Now, second is spleen. It is a bean-shaped organ. It is a bean-shaped organ, which is the largest single mass of lymphoid tissue. Which is the largest single mass. Single mass means there is no, diverticulum. There is no part, okay, of it, no lobe of it. Single mass of lymphoid tissue. Single mass of lymphoid tissue. Okay. During fetal life, in fetus, the spleen produces all types of blood cells. During fetal life, spleen produces all types of blood cells. Sorry. So during fetal life, spleen is a erythropoietic organ. It undergoes or in which hemopoiesis happens. Okay. Now coming to next, macrophages of spleen are phagocytic. Macrophages of spleen are phagocytic. Okay. Then tonsils, tonsils, they act as a filters to protect body, okay, from different type of pathogens, okay. They help in the formation of white blood cells. Peyer's patches. Peyer's patches, they are the collection of lymph nodules. Peyer's patches. They are the collection of lymph nodules found in small intestine in the ileal part. And they produce lymphocytes. Okay, so secondary lymphoid organs, it includes lymph nodes, spleen, Peyer's patches, tonsils. Okay, I haven't discussed tonsils because I will be discussing this part tonsils when I'll make you understand about the pharyngeal region. Okay, now next is this is the lymphatic system, the Waldear's ring. I will make you explain when I will make you understood about the pharyngeal regions. Okay, lymph nodes, tonsils, adenoids. Okay, then bronchus-associated lymphoid tissue, lymph nodes, bone marrow, spleen, Peyer's patches, mesenteric lymph nodes. Okay, this is the bone marrow and thymus. They are the primary lymphoid organs, and rest are the secondary lymphoid organs. So, my dear students, this is the last mucosal associated lymphoid tissue. Mucosal associated lymphoid tissue is basically an aggregation of lymphoid tissue, which is seen associated with the mucosas of respiratory tract, alimentary tract. Okay, it makes 50%, more than 50% of the lymphoid tissue in the body. Okay, different categories like gut, okay, gut associated lymphoid tissue, bult, bronchus associated lymphoid tissue. So you can see Peyer's patches also belong to the mucosal associated lymphoid tissue. Okay, so these are the site of production of B lymphocytes, T lymphocytes. Understood? They also produce macrophages and dendritic cells. Okay. So this part is completed. So we will meet in the next turn when we discuss or talk about the different types of diseases, my dear students. Okay. Till then, bye bye. Take care. Stay safe. Stay happy. Bye.